very much for the kind introduction, and also it is a pleasure to be here and give a presentation to you about my favorite topic, multiple myeloma. I'm really honored to be here. I have never been in Poznan. I've been several times in Poland, but never here. So really, it's a nice place to visit. And I tried to organize my presentation around the theme that as a medical student, I learned about cell biology and glycolysis and citric acid cycle, oxidative phosphorylation as something that you need to learn, pass the exam, and never use it again. And I will show you by the end of the presentation that this is something you need to know forever, because many of our drugs that we use to treat multiple myeloma operate on the way of metabolism, and influencing metabolism is something really important. And I would like to start with a simple slide that all roads in the ancient world, they say, that led to Rome, and some of the roads I will try to describe in my presentation that leads to the better treatment of multiple myeloma. But I will start somewhere very far away, a long time ago. I don't know, ever heard of the Sirius Passet? It's a locality in northern Greenland, where you can find fossils from early Cambrian, the time of the Cambrian explosion, where multicellular life originated really and spread on Earth. And this is a locality where you can find a lot of fossils. And paleontologists are really good in getting a lot of information out of these fossils. And at that time, this was an area which was a shallow ocean, and 600 million years ago it was near to the equator, and life was very abundantly living there, the shallow ocean a couple of meters deep. And paleontologists are really good, and now they can use different isotopic techniques to judge what were the circumstances that organic life originated there. And most interesting for me was that it was now calculated that this was under hypoxic condition. So not like now we have 21% of oxygen in the air, but at that time it was only maybe 10% in the air and much less in the ocean, about 6 to 10 meters deep. So what we need to remember that most of our cells originate from these ancient animals that developed in an area of hypoxic conditions. So hypoxia is something normal for our cells. So when cancer cells gain new properties under hypoxic conditions, this is something they just need to remember from ancient life when the life originated on Earth. So what is the effect of hypoxia on the cells? In the case of hypoxia, of course, oxidative phosphorylation is not working properly because the oxygen is not present in sufficient amount, and as a consequence, the ratio of the oxidized and the reduced NAD is changed, and of course you have a lot of reduced NADH, and that will have a lot of important effects on the rest of the cell. Like, most importantly, LDH and the malic enzyme, which we learned, of course, in the citric acid cycle and glycolysis, will change their activity, and they will produce some abnormal metabolites, like they will use oxoglutarate instead of oxoglutarate. They will produce hydroxyglutarate. Of course, hydroxyglutarate is very well known for hematologists because of the acute myeloid leukemia. We have IDH inhibitors, and the IDH inhibitors, they work because they block the production of hydroxyglutarate. But hydroxyglutarate is produced under hypoxic conditions always. So this is something really important to remember. And of course, hydroxyglutarate is an important second messenger to bind p53. And again, p53 is something we all know about, and this is something really important for cancer and tumor biology. But hydroxyglutarate is known to inhibit its ubiquitination. So this means this is something really important. It's a crosstalk between mitochondria and the cell nucleus and the p53. This is something important. So in the hypoxic condition, the mitochondria will produce the second messenger, hydroxyglutarate, and that will change a lot of things. In this figure, I present you that demethylating of DNA and histones is also glutarate dependent. So hydroxyglutarate may be able to inhibit the activity of glutarate-dependent dehydrogenases. Okay, so, and also hydroxyglutarate is known to activate mTOR. Again, something, of course, in myeloma we don't use anymore, but there is at least one protocol, is called PACMed, which was originated, which has an mTOR inhibitor. So this is something important, and when you starve, of course, mTOR is activated. So this is, again, something you need to remember as something from basic cell biology on the path of treating cancer. And when you talk about mTOR and inhibition, and soon you will reach MYC. And again, MYC is something really important for us as physicians who treat myeloma, because MYC activity is very high in plasma cells, and in the case of high MYC activity, usually you have trouble, because these cells will be in a cell cycle more frequently, and they will be more resistant to therapy, and of course these patients will have, unfortunately, a shorter survival most of the time. Okay, so let's find a new path. This is about myeloma cells. Of course, I won't talk so much because Dominic probably presented you all the most important stuff about myeloma cells and immunotherapy. So I won't talk about immunotherapy at all. So multiple myeloma is characterized by, of course, plasma cell proliferation in the bone marrow, and unfortunately sometimes outside the bone marrow. And probably you also know that all cases of multiple myeloma initiate from benign monoclonal gammopathy. And nowadays we also know that these monoclonal gammopathies start at early age. So probably some people in this room already have initiated monoclonal gammopathy in their body. Because according to the yeah, unfortunately, yes. Some people, even in their 20s, would have initiated the step. But this is really, really a slow process. And above the age of 80, it will be very common. Above the age of 50, it is about 5% of the population who has monoclonal gammopathy, which is a benign condition. You can think about it as like colon cancer starts with a little point of tumor, and then steps by step it will get to the cancer during the multi-step carcinogenesis process. And of course, things happen in the MGUS cells, and then myeloma will develop with anemia, bone pain, infections, lytic bone disease. So this will be a deadly disease. Unfortunately, presently is not really curable, but we all think that some patients are already cured. We don't know. Of course, myeloma is a special disease. It's very good disease with respect to follow up, because these plasma cells produce monoclonal immunoglobulins that could be quantitated, and the ratio, the amount of the M protein that is produced is usually proportionate to the tumor burden. So this is something really helpful for us as clinicians. But it is also important to know that immunoglobulins are very large molecules, and production of immunoglobulins makes a great burden on the plasma cells, so they produce a lot of garbage immunoglobulins. And this strain is important because plasma cells are very highly dependent on proteasomes. Proteasomes are tiny organelles that have a main function to metabolize and degrade unnecessary proteins, and therefore multiple myeloma is a disease that is exquisitely sensitive to proteasome inhibition. Probably you know that, but this was just a reminder. And this is what many myeloma patients present with. These are the so-called punched-out lesions of the bones, and this is a lytic bone disease that can cause fractures, and this can be a debilitating disease. And of course, multiple myeloma is multiple, as you see the PET scan on the left side, so it is present in multiple bones. So this is a general disease of the human body, and occasionally the myeloma cells can survive outside the bone marrow microenvironment. And this is usually extramedullary growth is usually associated with highly active and more resistant disease, and usually of poor prognosis. Of course, multiple myeloma is not just multiple in the sense of present on multiple places in the human body, but it is also a complex disease. There are, we think at least there are 8 different subtypes of multiple myeloma based on the gene expression profile, and also based on the founding genetic events that underlie the disease. So, hyperdiploidism is usually representing half of the myeloma cases, and the other half are presenting with a translocation that involve the immunoglobulin heavy chain translocation. And most common partners are cyclin D1, MMZ, and MUF. And they present as different types of myeloma, and they do present different sensitivity towards the different anti-myeloma agents. Targeted therapy of multiple myeloma started with the introduction of thalidomide. Of course, you all learned in the school about thalidomide embryopathy, and it's interesting to remember that thalidomide embryopathy was first noticed in the 1950s when this drug was introduced as an anti-emetic agent to be used in pregnancy. So this is something really interesting. And another thing important to remember about thalidomide, that thalidomide has no undesired effects in rodents. And this is something important always to remember, that sometimes really, really small changes may make a big difference. It's easy to study thalidomide and their analogs in animal models, like in fish and chicken embryos, because they have the same effect. So the chicken embryos will grow without wing if you add thalidomide or lenalidomide or pomalidomide or any of these analogs. But in mice, they have no effect, unless, of course, you genetically engineer mice which have the same tryptophan residue as in the humans and in other species. So, of course, by now we have genetically changed mice. And I think this is about the chicken. And okay, so how IMiDs work? So IMiDs are thalidomide analogs because we don't use so much thalidomide anymore in clinical medicine because of its toxicity, but we do use thalidomide analogs like lenalidomide and pomalidomide. And it has been known for a long time that these drugs have multiple effects on immune modulation, and that's why they called immunomodulatory agents. And for a long time, it wasn't known how is the mode of action of these drugs. And finally, using the animal models, it became clear that thalidomide and its analogs will work as modulators of the ubiquitin-dependent protein degradation. And what really happens in the presence of these molecules, there is a protein called cereblon, which is cereblon is there, which is ubiquitin protein ligase, and in the presence of thalidomide analogs, it changes its substrate affinity and substrates that may be ubiquitinated and degraded. And what is most important with respect to immunomodulatory effectivity is its effect on two zinc finger transcription factors, Aiolos and Icaros, and this will be most importantly responsible for its immunomodulatory effects and also its anti-myeloma effects. And this figure shows the importance of the tryptophan 388, which is a unique part of the binding site of the glutaramide site of the imide molecules on cereblon, which is different in mice than in all the other animals, including humans. And therefore, this is something really essential for its binding affinity. And of course, we use lenalidomide a lot. We use lenalidomide as a maintenance therapy after stem cell transplantation in myeloma. It is very useful. It is increasing the anti-myeloma immune response and also has direct anti-myeloma effects. And also, lenalidomide is used as a backbone of many, many therapies because lenalidomide has a synergistic effect of with most monoclonal antibodies and also proteasome inhibitors and steroids of anti-myeloma effects. So this is something really useful for us as clinicians, and we use this drug extensively. And fortunately now it's generic and it's not that expensive anymore, so it is not a great burden on the society. But there are multiple new IMiDs, and this is something, again, the drug development is ongoing, and this figure shows the activity of these thalidomide analogs on the cereblon. The way how ubiquitination of proteins is changing in the presence of the different drugs, having higher and higher affinity, lower and lower concentration is needed to achieve the effect. But there is more to that. It is not just the activity of these drugs are achievable at a lower concentration level, but they have a different effect on the cereblon protein and a different way how they influence its substrate specificity. This is a very good summary slide that shows that in the presence of these drugs, cereblon ubiquitinates some protein molecules inside the cell in the presence of each and every of these molecules. There are some substrates in the cell that are ubiquitinated and degraded in the presence of certain of these molecules, and there are some that are unique. Let's say lenalidomide is famous to be able to induce the degradation of casein kinase 1, and the other ones, they don't do that. This is important because this is a critical protein molecule that makes lenalidomide very active in myelodysplastic syndrome 5q-, and the other drugs don't work because they don't have this special effect on degradation of casein kinase 1, and so on and so on. So this is more to that the drugs have a good effect and an increasing effect on protein ubiquitination, but they have a different specificity on the substrate. So I usually explain it to my colleagues that this is kind of the cereblon takes a smell, which one of the IMiD molecule is present, and then changes its substrate specificity accordingly. This is something really important and interesting, and this can be used further in using novel molecules that have a different target. And the most novel two cereblon modulator drugs that are just entering the clinical practice are iberdomide and mezigdomide, and they have this unique ability to fix cereblon in one certain conformation because they have this long side chain that has an additional binding site on the cereblon and fixing it in the most active conformation. So they are very active in degrading Aikalos and Aiolos. And these are the two most important target molecules with respect to treating multiple myeloma. Again, a new path. So drug resistance. Again, as clinicians, we have the most trouble with the emergence of the drug resistance because we have multiple drugs, 8 or 10 very useful drugs that we can use to treat multiple myeloma, but sooner or later, all myeloma becomes resistant for the drug we are using. And what are the most important mechanisms that are involved in development of the drug resistance? Of course, if the gene is deleted, the target of the drug, then we have a problem because we have no target. Of course, loss of heterozygosy can aid in that. Point mutation of the drug binding site can happen. Probably Dominic already showed that in the case of immunomodulatory drugs, this is also a problem, that point mutations of the binding site are emerging as a problem, but it is also they are also emerging with the small molecules. And drug efflux mechanisms, they are a problem with the small molecules. The cells tend to pump out the drug. But the most important in the case of myeloma and drug resistance is the rearrangement of the signal transduction pathway. But the drugs we use that modulate a certain pathway and try to induce apoptosis of the myeloma cells, that somehow the myeloma cells learn to circumvent and maintain activity and avoid apoptosis. And of course, finally, especially with the drugs that are involving mitochondria, and this is something I will talk more later about, that mitochondria can be transferred between cells. So what we learned in cell biology, that the cell is the unit, I don't think so it's true anymore, because we of course know about extracellular vesicles and microtubules and nanotubules between the cells. So yes, cells are exchanging a lot of material, and this is something important with respect to tumor survival. So I already mentioned that protein accumulation inside the plasma cells is something that is deleterious for the plasma cells. So proteasome inhibitors are really a cornerstone of the myeloma therapy, and bortezomib was the first maybe 20 years ago when it entered the clinical therapy as a proteasome inhibitor, and somehow the cells are made in the presence of bortezomib to suffocate of unnecessary proteins. And of course, resistance is emerging against proteasome. Sometimes it's point mutation, but most importantly, these are additional alternative pathways to the cell to avoid protein overload and unnecessary protein, unfolded protein response. Of course, complexity is tremendous. I will jump through. And there are some newer generation proteasome inhibitors that are useful. They are in the clinic, like carfilzomib. We use extensively because it's an irreversible protease inhibitor, and it has multiple sites in the proteasome that can be blocked, so it is less sensitive to certain mutations that may emerge. So if you have multiple targets, then you are better off, but unfortunately multiple targets may have more side effects because carfilzomib has cardiac toxicity, and of course cardiac cells also have a lot of metabolism of proteins that need protein degradation, and if it is blocked, then you get cardiac failure. And sometimes surprises happen, like nelfinavir. This is an AIDS drug, which is a protease inhibitor, which is used, is not so much used anymore in HIV therapy, but it is present on the market. And it was observed that in the presence of nelfinavir, the unfolded protein response is changed, and much lower doses of bortezomib are enough to induce apoptosis of myeloma cells. So there is synergism of the drugs, and this is clinically helpful in some cases. Lenalidomide, we talked. Of course, lenalidomide has emerging resistance, especially since we use a lot of lenalidomide nowadays. And most importantly, cereblon expression decrease, but you can see on the slides that if the target molecule, cereblon, is decreased, then of course lenalidomide and all the IMiDs have less activity to induce myeloma cell death. Of course, there are possible point mutations, and the point mutations are not necessary in the cereblon molecule, but anywhere downstream they have an effect that may take away the effectivity of lenalidomide on myeloma cells. So we know a lot now about this resistant mechanism. Fortunately for us, it usually takes a long time for the myeloma cells to acquire these mutations, and especially these happen when the myeloma cells are treated with certain drugs that may induce mutations like melphalan. Again, what this figure tries to present to you to the critical importance of MYC in the life of the myeloma cell, and ICAROS and IOLOS, ECASAF and ECASAF3 as playing an important feedback role in MYC maintenance. And of course, I would like to draw your attention to the different possibilities of myeloma cells to downregulate cereblon and also delete of chromosome 3p, with chromosomal deletion that also may lead to resistance to lenalidomide and all the groups, all the members of the group. Again, a new path. Going back to mitochondria. So this is something that you can see in many textbooks showing a big mitochondrium and a small nucleus, and everything is shown in a circle. So these are obviously the wrong proportions. Myeloma cell may have about 500 to 1,000 mitochondria and one cell nucleus. So this is something closer to the proper proportions. So this is something to remember, that each and every cell have several hundred mitochondria. So mitochondria are really small as opposed to the cell nucleus. And mitochondria are important in medicine because the apoptosis processes that are initiated by mitochondria, and in that the anti-apoptotic protein, the BCL family members are really important. And we have a good drug, venetoclax, which is a BCL2 specific inhibitor, which is very commonly used in CLL, that is a well-known BCL2 dependent tumor. It's also used in acute myelogenous leukemia, but in certain cases of multiple myeloma, it has good activity. And venetoclax has very good activity in multiple myeloma with translocation 1114, which has cyclin D1 overexpression. And especially it has good activity in those myeloma cells that show B cell phenotype like CD20 expression. So it can be used as targeted therapy. It is very effective, especially it is used nowadays as a late-line therapy when the multiple myeloma cells, usually in Hungary it's used as fourth-line therapy, when all the other options in myeloma are exhausted, because this is still an off-label therapy. Everyone uses it, but still has no label. And it's very effective in translocation 11-14 and translocation 6-14. And this is, as I told, very effective in those cells which has a B cell type phenotype, CD20, CD79A positivity. But we don't know the best position of this drug in myeloma, because we may have a role for this drug in a broader area. And this shows that not all myeloma with the translocation 11-14 and cyclin E1 overexpression have sensitivity to venetoclax, but those which have overexpression of the B cell type phenotype, especially driven by the BATF transcription factor, these are usually very sensitive myeloma cells. And what I have seen in my clinical practice, sometimes you can get a response within a couple of days. Even tumor lysis syndrome in end-stage myeloma is possible. So this is something really important to keep in mind as a clinician. So how can we enhance the effectivity of venetoclax in myeloma cells? So myeloma cells are reliant on two most important metabolites for survival. They use glucose as food source, and they use also glutamine as food source. They need both, but they can survive on either of them. Of course, this is correlating somehow with that fact that obesity and impaired glucose tolerance is related somehow with the development of myeloma. But also the cells surviving glutamine withdrawal will have an increased effect of a BIM, which will bind to BCL2. But if we go back a little bit, this is something, again, to consider. If we have a higher amount of apoptosis-inducing BIM protein present, induced by glutamine deprivation, then you will have probably a higher effect of venetoclax, because more protein will be released from BCL2. This is something important as a clinician to consider, because how can you decrease the glutamine levels in your body? Physical exercise, because muscle uses glutamine. So what you can see that patients who exercise regularly and in better physical shape tend to survive longer. And this may be one of the effects of glutamine deprivation by muscle. Skeletal muscle uses a lot of glutamine, especially in the period after exercise recovery. It may take several hours, and the glutamine level in the blood may decrease by 70%. So this is kind of starving your tumor by exercise. Of course, this is something important, but of course, there are drugs in development which will inhibit glutamine uptake by the cells, like this drug which I indicated on the slide, which is also enhancing the effect of venetoclax. And oxidative phosphorylation, if you slow it down a little bit, like with metformin. Metformin is a common drug used in diabetes, and of course it's an OX-FOS inhibitor, and this enhances the effect of multiple different anti-myeloma agents, and this is something, again, important not to forget when you treat multiple myeloma. So, and a few words finally about our own research. We are doing, and we have been doing culturing of primary myeloma cells drawn from myeloma patient bone marrow. And it is usually difficult to culture myeloma cells, but we have seen that if you co-culture bone marrow stromal cells and myeloma cells coming from the same patient, they tend to survive very well. And usually if you co-culture myeloma cell with a feeder cell coming from another patient, sometimes they do well, sometimes they don't. So it's important to remember that since myeloma takes 15, 20, 30 years to develop, they co-develop with their bone marrow stromal cells, and they like each other, and they thrive in the presence of each other. But each and every one of them alone is not very easy to culture. We have about more than 250 different myeloma surviving cultures by now in our freezer. So we did some experiments, and what this shows that MSCs are the myeloma stromal cells. They are not influenced by carfilzomib, but what you can see that myeloma cells in the presence of stromal cells not only survive much better, but they have a decreased sensitivity to myeloma drugs. So somehow the stromal cells protect the tumor cells. And this is, I think, probably a quite general idea, because when you treat like chronic lymphocytic leukemia patients with venetoclax, you get a very fast clearing from the blood and very slow clearing from the bone marrow and the lymph nodes. So this is what we also see here in our co-culture system. And what we examined is that some part of this additional resistance to drugs may be due to mitochondrial transfer. Like stromal cells exchange mitochondria with the myeloma cell. And some of the mitochondria that are maybe influenced or damaged by the anti-myeloma agents, the stromal cells may uptake, and in exchange will give healthy mitochondria to the plasma cell. And this is something which you can see, you can visualize by confocal microscopy, that you can see traveling mitochondria in the tunneling nanotubules between the cells. And you can also label the mitochondria and study them. Here you can see that if you label mitochondria of the stromal cells and later plate plasma cells from the same patient on them, you can see that after getting physical contact in the presence of the drug, the mitochondrial exchange is enhanced. And may I have the short video? Oh. Okay, so what you can see here are mitotecal labeled cells, which are all the stromal cells, and plated on top of them are plasma cells. And this is a time lapse, multiple pictures taken, and you can see some of the plasma cells are getting red. And these are the mitochondria that were uptaken from the stromal cells on real time. So it is something interesting. Hop. Apologies.
Maybe one more time.
here are some more scientific figures in which we use flow cytometry to measure, in fact, the transfer of mitochondria and how they are influencing the survival of the cells in the presence of carfilzomib or venetoclax with respect to sensitive plasma cells. And what you can see here, that mitochondrial transfer has a good correlation with the survival of the plasma cells. So this is something definitely important to consider. And this is also can be observed from the viewpoint of ATP synthesis in the presence of carfilzomib. And in the presence of the stromal cells, the mitochondrial transfer will protect the ATP synthesis of the tumorous cells. And again, this is something related to better mitochondrial function gained by the additional mitochondria transferred from the stromal cell to the tumor cell. And this is something that leads to Dominic's presentation, that once the cells are becoming resistant to venetoclax, they are usually resistant to everything, because this is a late-line therapy, except the immunotherapies. The immunotherapies still work. So this is something, again, important when you think about scheduling the different therapies with respect to your myeloma patients. This is a usually incurable disease, especially in the relapse stage, so that you have to always, as a clinician, consider how is the best sequence to administer the available drugs. So immunotherapy usually does work after venetoclax. And this is, again, some of our own experiments. Belantamab mafodotin is an antibody-drug conjugate, so it's kind of an immunotherapy. And what we noticed that in our culturing system, it is not universally active. And but you can see that you're supposed to have each and every myeloma cell a lot of BCMA on the surface, but what we see is that some myeloma cells have very low expression of BCMA already. And these are myeloma cells that were never treated by BCMA agents and still have a very low expression. So unfortunately, some myeloma cells have very high level of BCMA positivity, and nearly all of the cells are positive. And unfortunately, we have seen some myeloma patients never treated with a BCMA agent has only 30% of the plasma cells having high expression. Maybe for immunotherapy, low expression is enough, because we know, like from T-cell lymphoma, that even if it is if it is CD30 negative, anti-CD30 may work. But again, this is something as a clinician important to consider. And again, we have noticed something interesting, that bone marrow stromal cells have zero BCMA expression, but in the presence of anti-BCMA belantamab mafodotin, we have seen an effect on mitochondrial transfer. So somehow the plasma cells that are damaged by the BCMA agent communicate with the stromal cells that please give me help. So it's interesting. It's interesting. We will. And this is my final slide, and I would like to thank you for invitation, and especially this is a national holiday in Hungary because of the October Revolution against the Russians and communism. It's today. Thank you very much. I'm happy to answer questions.
Professor Mikala, thank you very much for this fascinating talk. So I open the discussion. Please inform me if, in case you have also some questions from the audience on the internet. Yes. So, the first question, Professor Jaruzelski.
Okay, thank you. That was really interesting, and some molecular aspects. I'm a geneticist, so I appreciate such things. First of all, I was not aware that mitochondria can travel between cells. That is really great, and it can also take place in different contexts, physiological contexts, yes, I believe. Yes. Now, you mentioned that your myeloma cells... Yeah.
It's very interesting when there is blood clotting in your body ongoing. The little platelets usually get rid of the mitochondria because they kind of give it to the community.
That's great, really.
So it's interesting. So you can have the mitochondria floating in your blood at the time of blood clotting.
I see. I have another question. You mentioned that myeloma does not grow very well without stroma, which are from the same patient. Maybe the stroma, they are like a niche for myeloma cells, right? And maybe, as you mentioned, that this process takes many years, that there's a crosstalk between them, and this crosstalk is dynamic, and this both type of cells, they somehow learn how to interact. And in case you put it on stroma from another patient, they need to learn that again. Yeah. Maybe there's something like that.
And there are some results that showing that epigenetic changes occur in stroma cells. And maybe some myeloma cells rely a lot of interleukin-6, and some other myeloma cells rely a lot on insulin-like growth factor 1. But sometimes they are not interchangeable, and so on and so on and so on. So this is something can occur in a disease that takes such a long time to evolve in your body.
Exactly. Okay, thank you.
Further questions?
So congratulations on your research. Actually, I saw the movie some time ago about this mitochondria, but it's always fascinating. I have a question on the strategy and the future of the approach. So we did some research on the resistance to proteasome inhibitors, and we, in some extent, had similar results, like BCL2 may be involved in this resistance process. So in some extent, we had similar finding. But my question is the place for such strategical thinking to finding the drugs that may overcome the resistance to PI, like venetoclax, for example, or nelvinavir, for example, because I'm just thinking that probably the society will have the approach in the way that, for example, okay, we have resistance to bortezomib, we have some other drugs, let's forget about it and go forward. You think still there is a place for such overcoming the resistance to PI or lenalidomide, for example?
I would say, I will give you an example. If you have a plasma cell leukemia patient, primary plasma cell leukemia with 1114, would you use venetoclax in the first-line therapy? Because I would. Because this is something definitely a very high-risk disease when you don't want to develop very quickly resistance, so you want to add all the best available drugs in the first case. Probably you would think along.
Well, probably yes. We don't have that case, and we do not really check for 1114 on the regular basis. But
this is our protocol, that if you have a primary plasma cell leukemia, we have translocation 1114 done on the same day. And next day, we can decide whether we use aggressive chemotherapy or much less chemotherapy and venetoclax on the way.
Well, that sounds interesting. However, it's off protocol.
And very few patients are involved in this, couple of patients a year.
So still, this is a good idea to follow this path.
I understand the meaning of your question, that on the long run, probably newer classes of drugs are more important than salvaging the existing ones. But there is also an aspect of the existing drugs getting cheaper and cheaper and more available for the society. And if you have additional inexpensive drugs that you can salvage, so the society will have a good impact if you can figure out things. Not on the expensive novel drugs tend to be tremendously expensive and burden for the society. And if you can have two inexpensive drugs in combination being similarly effective, then especially for the developing countries of the world, and this is something important.
And still it's better to understand what's
Yes.
going on. Mhm.
Further questions?
So you showed that the presence of stromal cells reduces the effectiveness of drugs targeting multiple myeloma. So are there drugs, and is it like feasible, or would be a too big burden for the patient to like target the stromal cells to
Yeah. It's interesting
to,
that
to enhance the therapy?
many of the drugs we currently use have an effect on the myeloma stroma cells, like bortezomib and IMiDs. They all have an effect. And I think that the novel drugs, the bortezomib and lenalidomide, are especially have a good activity against myeloma because they not only target myeloma, but also the microenvironment. So I think off-target activity is sometimes very helpful.
So I have to some extent a follow-up question. So if there is a premalignant state, so I guess the cells might have some genetic lesions.
Yes.
So maybe there is an option to eliminate the cells long before the disease.
Yeah, currently research is ongoing on treating smoldering myeloma, which is a state between MGUS and symptomatic myeloma, especially with respect to high-risk smoldering myeloma, which is a disease that is expected to progress within a couple of years. Mm-hmm. But MGUS, I'm not really aware of any pharmacological attempts, but maybe dietary attempts. I think there are some ongoing studies for like intermittent fasting, which is now getting more popular.
We have sport at least, yeah?
Yeah, a sport. Yeah. But now you know intermittent fasting is like eating 6 to 8 hours a day and starving for the rest of the time may have an effect on MGUS. And there are some studies ongoing which are checking the M protein levels with intermittent fasting. Dana-Farber Institute is running a trial like that. We don't know. We will learn in 10 years whether it's effective.
But there are studies showing that low physical activity is related to the higher
yes, and obesity is related. Yes, so diabetes is related. I mean.
So that's why I'm going to the gym and the.
Yeah. And starving your myeloma cells. Yes.
Everybody should, right?
Yeah.
Yes, Susan?
Here. You mentioned that in some cases when venetoclax resistance occurs, you can use CAR-T therapy or immunotherapy for multiple myeloma. Can you please tell me how often that happens and what is the effectivity of this immunotherapy?
I don't know.
Okay.
I don't know, unfortunately. Probably there are occasional cases, but I'm.
It's given, just out of protocol, in just relatively rare situations. So there are no set observations. At least I can follow this.
We can check the literature. Maybe there are cases, but I'm not aware. No.
Do we have some questions from the internet audience? Not. So, if there are no more questions, so let us thank again all our today's speakers for their absolutely excellent presentations. And I also like to thank the audience here in our seminar room, but also on the internet. And of course, our sponsors for supporting us financially. And taking this opportunity, I would like to invite you already to the Hematology Day 2025, which will be organized also in autumn next year. And then we will focus on skin lymphoma. So please all be invited, and thank you for today's vivid discussion and the presentations. Thank you a lot.